Hello everyone, I am Sriagilandeswari Ravichandran from Department of Bioinformatics, Shastra University, India. My poster talk is about detection of cryptic site with molecular dynamics data using machine learning technique. First of all, cryptic site. It is a hidden binding pocket on protein surface that remains undetected in static structure but accessible under certain conditions such as conformational changes. It is important to detect cryptic site because many undruggable proteins became druggable by detecting the cryptic site and targeting that particular region. So this basically allows for improved specificity and reduced toxicity, which is a very attractive novel drug design strategy. Here, we mainly focus on integrating molecular dynamics and machine learning techniques because critic sites remains hidden in static structures. So here we took 19 protein PDB ID and chain identifiers for Um, cryptosite analysis here web server which identifies residues in the protein is cryptic or not with with the faster sequence we did conformational sampling in collabfold 2 which has two strategies. One, activating dropout to increase uncertainty in the model, and the second strategy is to reduce MSA depth, which reduces the number of sequence used to increase diversity. With the MD data, we basically did RMSF per residue plot, and from that the peak regions were taken for further SASA analysis, where increasing trend residue regions were taken for comparison between cryptosite analysis results and these regions. And we did PyEMA, which is a package, Python package, was used to analyze the MD data. And now we can discuss the results and discussion. Here, this is the 19 proteins we have taken, and this figure 1 is the result of cryptosite analysis. So, this blue region is nothing but non-cryptic residues, and red is cryptic residues. And the next is validation of cryptic site from molecular dynamics simulation data. So we did RMSF per residue, and then the peak region is taken for further SASA analysis, and then the regions with increasing trend is taken from this analysis and then compared with the cryptosite analysis, and here the residues were listed for flavin reductase protein, and conformational sampling was done using collabfold 2. So in collabfold 2 we actually did all the two strategies. So blue colored one is MSA depth reduction, and orange color is nothing but the dropout activation strategies. So here I have marked the maximum RMSD we are getting from the 16 iteration of each strategies, and then PyEMA was done. So for this we have to extract features from MD data. For each protein we have to do a separate PyEMA program. So the features taken were coordinates and tangent angle, and PZA was done. K-means clustering algorithm was used to calculate to cluster the similar structures into 100 cluster groups. Markov state model was used, and n states were assigned using eigenvalues and identified which cluster group present in which state. So this plot is nothing but reactive network plot, and then TPA, transition transition pathway analysis plot. So the plot A is a reactive network plot where nodes are MSM state and edges are the time when the transition from one state to another happens, and the thickness of the edges is proportional to the transition probability. So here flavin reductase has denser network. The predominant transition occurs from state 4 to state 3, and for the plot B, you can see that this plot is transition pathway analysis, where we'll be getting where the pathway occurs from state A to state B, and the committer probability is on the x-axis, which indicates how likely a state is to reach A before B. And uh, and here, um, the features were taken from MD data using Python code with the package called MD Dredge. So the features were SASA, hydrophobicity, RMSF, phi-psi entropy, charges of the residues, and density across neighboring residues, like across phi Armstrong. And secondary structure were extracted from MD data using the MD Dredge pack, and class label was assigned as 1 for residues that has increasing trend in SASA analysis. Otherwise, all other residues will be 0. And the data set of 19 proteins were built, and there is a severe data imbalance. We can see the plot 5, where class distribution is explained, and I have used borderline mode to balance the data set. And then 7 algorithms were used to train the model. Like for 80 percentage of the data were trained, and then 20 was used for testing. And I have got the good result. Like XGBoost has the best performance, and like in the performance graph, like in ROC curve, the diagonal gray line represents the random guessing. So the closer the curve is to the top left corner, the better the model. And for the PR curve, the closer the curve stays to the top right corner, the better the performance. So for the conclusion, integrated dynamic analysis for critic site detection. This study actually uses molecular dynamics data and then static structures. And with the help of these two, we have done the we have detected the cryptic site, and we have successfully integrated the molecular dynamics and machine learning framework. And thank you.